start today's episode, these are the 5 wonder tips for you to remember to keep your back, neck, and eyes healthy while indulging in some screen time. This is Teacher Melvin's healthy tips for everyone. Number 1. Sit properly so you are well supported. Your back should be touching the chair backrest. Number 2. Change your position regularly. If you are watching for long periods, take turns sitting on the couch, chair, or floor. Number 3. Keep dim lighting on in the room rather than no lighting. Strong or no lighting may lead to eye damage over time. Number 4. Stretch. Use commercial breaks to change positions, to get up and walk around, and to do gentle stretches. Number 5. Don't watch TV in bed, as this often leads to neck and backache. There, you are now ready to begin. Hello, senior high school STEM learners! Welcome to our biology class, where learning never stops and the adventure never ends. So buckle up and get ready to power your mind. I am Teacher Melvin, and this is Wonder Science. Have you ever seen a caterpillar turn into a butterfly? If your answer is yes, you are probably familiar with the idea of a life cycle. Butterflies go through a spectacular life cycle transition, turning from something that looks like a worm into a pupa, and finally into a beautiful creature that floats on the breeze. A life cycle is a series of developmental steps that an individual goes through from the time it is born until the time it reproduces. The cell cycle can be thought of as the life cycle of a cell. What is cell cycle? Does it ring a bell? For today's episode, we will learn more about the cell cycle and why it is thought as the life cycle of a cell. Are you ready? To begin our learning exploration, let's have a short game. For this activity, you will need the following materials: a pencil and a piece of paper. Do you have the materials now? If so, let's begin. Here are the mechanics of the game. First, I will show you 5 words with jumbled letters in them. Second, quickly unjumble letter combinations to form the correct word. Arrange the letters as fast as you can because you are only given 60 seconds. When you hear this sound, that means time is up. For every correct answer, a Wonder Badge will be given to you. Collect all 5 to get your first Wonder Gold Medal. Get ready because the game will begin in 3, 2, 1, go! Time is up! Let's check your answers. These are correct words from the letter combinations: prokaryotic, eukaryotic, cytoplasm, organelles, centrosome. There! How many words have you correctly arranged? If you got all 5 Wonder badges, here is your first Wonder Gold Medal! Earlier, I have mentioned that the cell cycle can be thought of as the life cycle of a cell. In other words, it is the series of growth and development steps that a cell undergoes between its birth, or the formation with the division of a mother cell, and reproduction, or the division to make 2 new daughter cells. To divide, a cell must complete several important tasks. It must grow, copy its genetic material, and physically split into 2 daughter cells. Cells perform these tasks in an organized, predictable series of steps that make up the cell cycle. Well, what exactly is a cell cycle? Cell cycle is the name we give the process through which cells replicate and make 2 new cells. In eukaryotic cells, or cells with a nucleus, the stages of the cell cycle are divided into 2 major phases: interphase and the mitotic phase. During interphase, the cell grows and makes a copy of its DNA, or deoxyribonucleic acid. During the mitotic phase, the cell separates its DNA into 2 sets and divides its cytoplasm, forming 2 new cells. Let's enter the cell cycle just as a cell forms, by division of its mother cell. What must this newborn cell do next if it wants to go on and divide itself? During interphase, the cell undergoes normal growth processes while also preparing for cell division. In order for a cell to move from interphase into the mitotic phase, many internal and external conditions must be met. Interphase occurs in 3 stages, namely G1, S, and G2. What happens in each phase? Let's discuss one by one. The first stage of interphase is called the G1 phase, or the first gap, because from a microscopic aspect, little change is visible. However, during the G1 stage, the cell is quite active at the biochemical level. The cell is accumulating the building blocks of chromosomal DNA and the associated proteins, as well as accumulating sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. Second, the S phase, or synthesis of DNA. Throughout interphase, nuclear DNA remains in a semi-condensed chromatin configuration. In the S phase, DNA replication can proceed through the mechanisms that result in the formation of identical pairs of DNA molecules, or sister chromatids, that are firmly attached to the centromeric region. The centrosome is duplicated during the S phase. The 2 centrosomes will give rise to the mitotic spindle, the apparatus that orchestrates the movement of chromosomes during mitosis. Take note, centrioles are not present in the centrosomes of other eukaryotic species, such as plants and most fungi. And third, the G2 phase, or the second gap. In the G2 phase, the cell replenishes its stored energy and synthesizes proteins necessary for chromosome manipulation. Some cell organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the mitotic phase. There may be additional cell growth during G2. The final preparations for the mitotic phase must be completed before the cell is able to enter the first stage of mitosis. To sum it up, the G1, S, and G2 phases together are known as interphase. It is worth to note that the prefix inter means between, reflecting that interphase takes place between one mitotic phase and the next. Wonder learners, how are you so far? Remember, if you don't take notes, you will forget everything we discussed today. So make sure to write down important details for easy remembering. Earlier, you learned that the first major phase of the cell cycle is called the interphase, which is divided into 3 stages. Can you quickly recall the 3 stages? Excellent work! The three stages of the interphase include G1, S, and G2. Wonder learners, I commend you for your quick remembering. Keep up with the good work. And now, let's proceed to the next phase of the cell cycle, which is the M phase. The M or mitotic phase is a multi-step process during which the duplicated chromosomes are aligned, separated, and moved into two new identical daughter cells. Mitosis is the tightly regulated process of cell division that includes both nuclear division and the division of cytoplasm to two daughter cells. There are two portions of the mitotic phase. The first portion is called karyokinesis, or nuclear division, and the second portion is called cytokinesis, or the physical separation of the cytoplasmic components into the two daughter cells. In mitosis, the nuclear DNA of the cell condenses into visible chromosomes and is pulled apart by the mitotic spindle, a specialized structure made out of microtubules. Mitosis takes place in four stages: prophase, sometimes divided into early prophase and prometaphase, metaphase, anaphase, and telophase. The products of mitosis are two daughter cells that have identical DNA content that is also identical to the DNA content of the original parental cell. We will know more about the distinct phases of mitosis in our next episode. In cytokinesis, the cytoplasm of the cell is split in two, making two new cells. Cytokinesis usually begins just as mitosis is ending, with a little overlap. Cytokinesis takes place differently in animal and plant cells. In animals, cell division occurs when a band of cytoskeletal fibers called the contractile ring contracts inward and pinches a cell in two, a process called contractile cytokinesis. The indentation produced as the ring contracts inward is called the cleavage furrow. Animal cells can be pinched in two because they are relatively soft and squishy. On the other hand, plant cells are much stiffer than animal cells. They are surrounded by a rigid cell wall and have high internal pressure. Because of this, plant cells divide in two by building a new structure down the middle of the cell. This structure, known as the cell plate, is made up of plasma membrane and cell wall components delivered in vesicles, and it partitions the cell in two. What happens to the two daughter cells produced in one round of the cell cycle? Well, the answer depends on what type of cells they are. Some types of cells divide rapidly, and in these cases, the daughter cells may immediately undergo another round of cell division. For instance, many cell types in an early embryo divide rapidly, and so do cells in a tumor. Other types of cells divide slowly, or not at all. These cells may exit the G1 phase and enter a resting state called G0 phase. In G0 phase, a cell is not actively preparing to divide. It's just doing its job. For instance, it might conduct signals as a neuron or store carbohydrate as a liver cell. G0 is a permanent state for some cells where they remain metabolically active but no longer proliferate unless called on to do so by appropriate extracellular signal, while others may restart division if they get the right signals. In contrast to the rapid proliferation of embryonic cells, some cells in adult animals cease division altogether. Example, nerve cells. And many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. Cells of the latter type include skin fibroblasts, as well as the cells of many internal organs such as the liver, kidney, and lung. Did you know that different cells take different lengths of time to complete the cell cycle? The duration of these cell cycle phases varies considerably in different kinds of cells. For a typical rapidly proliferating human cell with a total cycle time of 24 hours, the G1 phase might last about 11 hours, S phase about 8 hours, G2 about 4 hours, and M about 1 hour. But fast-cycling mammalian cells, like the ones that line the intestine, can complete a cycle every 9 to 10 hours when they're grown in culture. Other types of cells, however, can divide much more rapidly. Different types of cells also split their time between cell cycle phases in different ways. In early frog embryos, for example, cells spend almost no time in G1 and G2 and instead rapidly cycle between S and M phases, resulting in the division of one big cell, the zygote, into many smaller cells. So there! You have just learned about the phases of the cell cycle, their characteristics, and their control points. You are now ready to move forward to the next lesson. But before that, let's evaluate what you have learned so far from our biology lesson. Challenge yourself to do this 5-item quiz. I will read the question and choices once. You're only given 5 seconds to write the letter of the correct answer for each question. You will be given 1 Wonder Badge for every correct answer. Are you ready? Wonder STEM learners, let's play Science Pop Quiz. Question number 1. Most of the life of any cell is spent in a period of growth called blank. A. Telophase, B. Prophase, C. Interphase, or D. Anaphase. The correct answer is letter C. Interphase. Question number 2. Which choice best describes the cell cycle? A. Cells grow and develop during interphase. Cells reproduce during the mitotic phase. B. Cells grow and develop during the mitotic phase, and cells reproduce during interphase. C. The nucleus of a cell divides during interphase, and the cytoplasm of a cell divides during the mitotic phase. Or D. The nucleus of a cell divides during the mitotic phase, and the cytoplasm of a cell divides during interphase. The correct answer is letter A. Cells grow and develop during interphase, and cells reproduce during the mitotic phase. Question number 3. How long does it take for most dividing human cells to complete a cell cycle? A. A few minutes. B. A day. C. A year. Or D. About 7 years. The correct answer is letter B. A day. Question number 4. During which phase of interphase do cells copy their DNA? A. S phase, B. G1 phase, C. G2 phase, or D. Cytokinesis. The correct answer is letter A. S phase. Question number 5. Which best describes how a plant cell divides? A. A new cell plate and wall forms in the middle of the cell and 2 new cells are formed. B. The 2 cells twist apart. C. The membrane pinches shut in the middle of the cell and the cells are split apart. Or D. Plant cells do not divide. The correct answer is letter A. A new cell plate and wall forms in the middle of the cell and 2 new cells are formed. Excellent job, STEM learners! I am happy that you were able to challenge yourselves to do the pop quiz. Now, let us add all the wonder badges you've collected from the pop quiz. How many badges have you collected? For all the effort that you do, I will give you your second Wonder Gold Medal. For your assignment, perform this activity at home. Arrange the following events in correct order. Spindle fibers emerge from the centrosomes. Chromosomes condense and become visible. Nucleolus disappears. Nuclear envelope breaks down. Write your answer in your biology interactive notebook. And that ends our Wonder Learning exploration for today. But don't worry, more science adventures are coming your way. Keep exploring and never stop learning. Always remember, learning biology is fun for everyone. This has been your Wonder Teacher Melvin. See you next time for another episode of Fun Wonder Learning Adventures. Only here at DepEd TV.